we have our final speaker, Alex Tyshkovskiy, from Vadim's lab. Let me find your presentation. Did I pronounce your name?
Yeah. Better than my parents. Thank you.
Thank you so much.
Dear colleagues, it's a great pleasure to be here, and thanks to Morten and Alex for this amazing event. Today we'd like to present you our work in Vadim Gladishev's lab on development of transcriptomic clock of biological age and lifespan in rodents. And I was really amazed at this conference how different are approaches of different labs, different theories of aging. But there is one thing we share in common, and that's we have ultimate goal to find a fountain of youth. And of course, in our lab, we're also interested in that. And the great instrument that all of us use is, of course, epigenetic clocks. And Steve Horvath gave a brilliant talk today. I'll just briefly highlight that currently epigenetic clock allows us to accurately predict phonological age of different mammals. And also, it can be applied to predict the effect of long-term lifespan extending interventions. For example, we observed that in response to caloric restriction and growth hormone deficiency, the epigenetic age of mice was indeed decreased. However, there are some challenges with epigenetic clocks, and some of them are listed here. It's sometimes hard to functionally interpret the results we observed. We see the effect, but what's behind it? Second, in terms of short-term interventions, epigenetic clocks are sometimes not sensitive. So it usually takes time to remodel the epigenome. And finally, now it's really hard to obtain DNA methylation data from single cells. So that's why it's hard to investigate the heterogeneity of biological age across different cells. So we decided to use other types of omics data to solve these issues, and we chose gene expression. Since there is tons of data available on gene expression, we aggregated data from 85 independent datasets on more than 2,500 samples of mice and rats across 26 tissues. And you may see that basically the whole lifespan of mice is pretty nicely covered by our data. But besides that, we were also interested to build clock that would not only predict chronological age, but predict biological age. We know we have data that some aging-related changes are not detrimental, but they can be neutral or even beneficial. So we want to separate detrimental changes from other changes. That's why we also included data on different models of mice, different interventions, genetic, pharmacological, dietary, etc., that can either extend lifespan or shorten lifespan, or do not change it at all. So our great collaborator, Rich Miller, provided us with data on 24 compounds from ITP studies. Some of them extended lifespan of mice, some not. And using all this data, we calculated lifespan-adjusted age, which is just the chronological age divided by the expected maximum lifespan of this particular model. And then we also looked that basically almost the whole lifespan is covered with our data, and you may see that we have different strains, genetic background strains of mice, we have different interventions, different types, so we try to get as wide as we can. Long story short, we developed a clock. We did it for individual tissues such as liver, muscle, and brain, and also we developed multi-tissue clock. And first model that predicted the chronological age, absolute age of mice, had a correlation of 0.86 on test set, which is lower than for epigenetic clocks, but it's still nice. However, we know that gene expression data is often accompanied with great batch effect. So how can we remove it? We decided to have, make a trick. Instead of predicting absolute age, we predicted the difference of age compared to randomly chosen control group for every sample. So in this way, we removed batch effect between gene expression profiles, and we were able to improve our clock. So now it has the accuracy of 0.94, which is comparable with epigenetic clocks. And this is for multi-tissue data. For individual tissues, the accuracy is even higher. And now I want to show you the difference, highlight the difference between chronological and biological clocks. So Steve Horvath mentioned today that epigenetic clocks sometimes fail to predict the effect of Hutchinson-Gilford progeria. We know that mice that have this syndrome, they live 10 times less on average than normal mice, but still for some reason many clocks fail to see the difference. And when we applied our transcriptomic clock, with chronological clock, we also didn't see the difference. So these mice were 3 months of age in both cases, and you may see that on average clock predicted 3 months. However, lifespan adjusted clock could see the huge difference between these models, and indeed predicted progeria mice to be biologically older. Okay, so now let's go to applications. First, we decided to validate our clocks using established models, which we used to validate epigenetic clocks. And of course, we started from aging-related diseases. So we aggregated data for different diseases, and for the majority of them, almost for all of them, our biological clocks indeed predicted higher age in diseased mice. So this is the data for ischemic stroke, this is for Alzheimer's disease, this is for type 2 diabetes, chronic kidney disease. The only disease that our clock failed to predict higher age was cancer, in this case hepatocarcinoma. And we know that cancer is not very typical age-related diseases. Some features of cancer cells may mimic features of young cells, so that's why probably our clock failed. Anyway, for the majority of disease, it worked. And the beauty of gene expression clock is that we can directly see which particular genes and functions are responsible for the effect we observe. And so we did functional enrichment, and we saw that many features, aging-related features of diseases are shared. So for example, we observed the upregulation of apoptosis-related genes, upregulation of KRAS signaling, and of kappa-B signaling, etc. We then turned our attention to the next model, which is early embryogenesis. And previously in our lab, my colleagues showed that during early embryogenesis of mice, we can observe the decrease of epigenetic age, and only then biological age starts increasing. So we decided to reproduce it using gene expression data, and we observed exactly the same pattern. So approximately between days 7 and 10, we see the minimum of biological age, which was called by Vadim Gladyshev as ground zero. And again, we can take a look at functions responsible for that. And we may see that some functions are responsible for both decrease of age in the early embryogenesis and increase of age afterwards. For example, adipogenesis, hypoxia, mTOR signaling, all of them show this U-shaped curve. And now the favorite model of all postdocs, which is sleep deprivation. So we took young mice and applied them to sleep deprivation for 5 days, 10 days, and also we had a group that had sleep deprivation for 10 days and then recovery for another 5 days. So they could sleep as they wanted to. And surprisingly, we observed very clear difference. So following sleep deprivation, we observe the increase of age, but then after recovery, it goes back to the normal state. We also used the same samples for epigenetic clocks. But for the epigenetic clocks, we saw the same trend, but it was not as clear. So as I mentioned before, epigenetic clocks are not always sensitive in detecting very short-term interventions. And here it's just 10 days, so it's not very long-term. Gene expression clocks can see the difference. And in this particular case, almost the whole effect was related to inflammatory response, which we know is indeed related to sleep deprivation. Now let's go to single cell level. So we decided to validate our clock and check if it would work on single cells. We used tabula muris dataset that has a huge range of tissues, and they tested gene expression of single cells across mouse lifespan. And we applied our bulk clock using pseudo-bulk technique. So we just randomly chose 500 cells, put gene expression data, and applied our clocks. And we observed that almost for all tissues, our clock was accurately predicted chronological age. Correlation was between 0.8 and 0.96. On average, it was 0.83 when we put all the tissues together. So knowing that, we can now apply our clock to single-cell data for different biological models. And we decided to do it for reprogramming. We know that OSKM reprogramming rejuvenates epigenetic age. It was shown hundreds of times. And we saw the same using our gene expression clock. So this is the data for reprogramming for mice and humans. For this reason, we also had to develop clocks for humans. But anyway, and interestingly, you may see that for mice, rejuvenation takes much less time. And this is consistent with the duration of reprogramming. So using original OSKM design treatment, for humans, it takes about 24 days. For mice, about 2 weeks. So, and we observe the difference also in terms of rejuvenation. Then we used the data from Broad Institute. They performed single-cell sequencing for cells during reprogramming, 250,000 cells. And they saw different trajectories of cells, different cell types. We applied our clock to this data, and we saw that there is some heterogeneity of transcriptomic age. And interestingly, higher transcriptomic age was observed in stromal cells. Even more interestingly, when we applied gene expression signatures of some functional processes, we observed that the same stromal cells have also upregulated level of apoptosis and senescence associated secretory phenotype. We applied trajectory method to see how transcriptomic age changes in different cell types. And we observed, again, high heterogeneity. In some cells like iPS cells or neural cells, we observed low average epigenetic, gene expression age. However, in our cells, like stromal cells, we observed it high age, and it was even higher than in original cells. And again, it was very well consistent with the behavior of apoptosis and senescence associated signatures. So not surprisingly, when we looked at correlation between transcriptomic clock predictions and different features of these cells, we observed high positive correlation with features of apoptosis and senescence, and negative correlation with cell cycle and pluripotency. So using this clock, we may see that also on average, we observed decrease of age during reprogramming. In fact, different cell types have very heterogeneous transcriptomic ages, and that's quite impressive. Finally, we decided to use the same data to build the clock of lifespan. Since we have different models with different expected maximum lifespan, we decided to build a clock that would predict not age, but lifespan, irregardless of age. And we were able to do so. The average correlation is 0.76. The clock was able to correctly predict higher lifespan for dwarf mice, both little mice and Ames dwarf mice. Finally, to see if we can apply it to predict novel interventions, we took human head mice and took 25 compounds and subjected these mice to these compounds for just 1 month. After that, we sacrificed mice and took liver and kidney tissues and applied our clocks. Our clocks predicted some of the compounds to be significantly to have significantly higher lifespan compared to control. And then some of these compounds were tested for their effect on health span and lifespan of old mice. This work was done by Anastasia Shindapina. She did a huge amount of work. And what she did, she took 25-month-old mice, really old mice, and applied 6 compounds listed here. All of them had improved lifespan according to our clock. She did it until their death. And she checked different features such as frailty index, gait speed, and also, of course, lifespan. So we observed, and this is some data for some of the compounds. For example, compound number 1, top compound, you see that it decreases both frailty index and improves performance. Also, finally, when we checked lifespan, we observed that 3 out of 6 drugs we predicted indeed significantly extended lifespan of old mice. 3 other compounds did not succeed, but this was performed only on old mice, so this is pilot study. Now we are going to try it for like the whole lifespan on young mice, since usually we observe that the effect on young mice till the end of their lifespan is usually higher. So to conclude, our gene expression clocks are able to accurately predict both chronological and biological age, and also, more importantly, characterize specific functions responsible for that. It can be applied to different established models of rejuvenation and aging. I didn't list all of them here, but we also have data on parabiosis, caloric restriction, etc. It can be used to look at heterogeneity across individual cells. And finally, clocks of lifespan can be used to predict novel interventions that could extend health span and lifespan of animals. So with that, I hope eventually all of us will be able to find a fountain of youth, and our lab will also play some role there. I would like to thank all our lab, Dr. Vadim Gladyshev, all our postdocs, and also all our great collaborators, including Richard Miller, Vera Gorbunova, Dragana Rogula, etc. Thank you for your attention.
Thank you so much, Alex. We have time for maybe one question. There is one from Joris. Run.
Hi, great talk. So when you analyze the single cell data from tabula modus, did it matter if you take the single cell data from 10x and SmartSeq 2? Did it change anything in your prediction because of the depth of the analysis?
We didn't do it yet. We will also check it later. For now, I have showed you just some pilot studies related to single cell analysis. We want to develop it even further and check different models, different platforms, etc.
So great talk. Did you already try to apply your clock to a human data set? Can you do a one-on-one translation? Because you mentioned shortly something about a human clock you created. Do you really need to create it differently, or can you try to apply the same clock to humans given the translational aspect of gene expression we normally see?
That's a great question, and basically initially we developed independent clocks for rats, mice and humans, and saw how well they correlate to each other. We observed that on average there is positive correlation, but the accuracy is not perfect. Especially it's not perfect in predicting the absolute value of age. So if we apply mouse clock to human data, we usually see positive correlation, but the real predicted values are just chaotic. They do not mean real values.
All right. Maybe I can also ask a question. So all these clocks are trained on the age of the animals. Have you thought about training it on the remaining time left until death? Because in many of the cases you will know how much time the mice has left.
Yeah, so in a way the clock that is predicting lifespan adjusted age is predicted to remain in lifespan, but in proportional values. So it predicts that like this mouse has remaining 40% of lifespan, so in percentages. But also, I didn't mention, but the clock that predicts lifespan, we did it in two ways. We first developed it just regardless of age. So the model didn't know the real age of mice. But second version, we developed the lifespan clock that would use both gene expression data and the chronological age data. And that clock was even better. So like the correlation was 0.8 or something like that. So if we also include the data on the real chronological age, it even further improves the prediction of remaining lifespan.
Very cool. Thank you so much.
Thank you.